Very good morning, students. Welcome to biology classes. Today we are going to see 11th science biology and we are going to see the next topics of chapter number 10, that is cell cycle and cell division. We have already completed with the first type of cell division, that is mitosis, and today we are going to see the second type of cell division, that is meiosis. Meiosis is also known as reduction division, right? And first we will see about the basics of meiosis and then we will shift to the different phases in which different things occurs, right? So meiosis is a type of cell division that results in four daughter cells, each with half the number of chromosome of parent cell as in the production of gamete. This type of division is known as meiosis. So here in meiosis, when we were talking about mitosis, two daughter cells were produced, but here in meiosis, four daughter cells are produced. And this four of the daughter cells is going to have half the number of chromosomes of the parent cell. So the number of chromosomes which is present in the parent cell, half of that is going to be present inside the daughter cells. And this is going to occur in the gametes, right? That is the gametic cell. So none another but this type, that is the meiotic type of cell division, occurs in the gametic cells. And upon the occurrence or upon carrying out the meiotic cell division, four daughter cells are produced, right? Now we are going to see about the key features of meiosis. So meiosis involves two sequential cycles. So in the meiosis, two cycles run, right? The first cycle is said to be the meiosis 1 and second cycle is known as meiosis 2. But only a single cell of DNA replication takes place, right? So here what happens in the meiosis, even though the meiosis 1 and meiosis 2 two cycle is running, but this two of the sequential cycle is occurring in single cell of DNA replication. Now what exactly meiosis 1 is? So meiosis 1, that this type of cell division starts when the parental chromosomes are replicated. So when the parent, whatever chromosomes which are present in the parental stage or in the parental cell, right? That parental chromosomes, they start the replication. So in the meiosis 1, the cell division takes place of the parental chromosome, right? And upon cell division, it produces identical sister chromatids. So the product of the first meiosis, that is meiosis 1, is going to be identical sister chromatids, right? So upon carrying out the first meiosis, that is meiosis 1, two sister chromatids are produced. So upon the division of the parent cell, two cells are formed. So in the meiosis, the product of meiosis 1 will be sister chromatids and that will be identical in nature. As well as the product, that is the number of cells, that is the parent cell was 1, but the sister chromatids which are formed in identical manner, they are 2. So meiosis 1's product, that is the product of the first cycle, is 2 cell and those two daughter cells are identical sister chromatids. Pairing of homologous chromosome occurs in meiosis. Now here what happens is the homologous chromosomes, homologous, homo means same, so the chromosomes which are same, like they are exactly similar with each other, right? Thus the pairing of those type of chromosomes takes place in the meiosis. This does not takes place in the mitosis, because here when we are talking about meiosis, as this type of cell division takes place in the gametic cell, for this reason the pairing of the homologous chromosomes takes place. Four haploid cells are formed at the end of meiosis 2. So two cells are produced at the end of meiosis 1, and from these two cells another two cells are formed, and for this reason after the occurrence of the meiosis 2, again two cells are formed. So from one cell, that is from the one parent cell, four haploid cells are produced. So the product of meiosis 1 will be two sister chromatids, and another, after the occurrence of the meiosis 2, totally four haploid cells are formed. So now this you can see is in the meiosis 1, the phases which are involved are prophase 1, metaphase 1, anaphase 1, and telophase 1. Same way, the cycles which are running in the meiosis 2, the phases it includes are prophase 2, metaphase 2, anaphase 2, and telophase 2. So now we are going to see all the phases in detail. So let's start with the meiosis 1, that is the first sequential cycle, and we are going to see about the meiosis 1. So in meiosis 1, the first phase which is coming, that is the prophase 1. This phase is complex phase which is compared to the prophase of mitosis. So comparatively, the prophase is more complex in the meiosis 1 than in the mitosis, right? So this meiosis is quite complex than the mitosis. So it is subdivided into 5 phases. So prophase 1 over here, it is divided into 5 phases. The first is leptotene, second is zygotene, third is pachytene, fourth is diplotene, and fifth is dikinesis. So these are the 5 phases of the first phase of meiosis 1, and that is prophase 1. So in the leptotene, what happens? The chromosomes are visible under microscope. So you cannot see through your naked eyes, but the chromosomes, when you are visualizing them under the microscope, you can easily see them, right? Compaction of chromosomes occur in the first phase of the prophase 1, and that is leptotene. So condensation of chromosomes starts to occur in the leptotene phase. Second is the zygotene phase. In the zygotene, the chromosomes pair with each other, and this process is known as synapsis. So in the second phase of prophase 1, that is zygotene, the chromosomes, they, as you know, that the homologous pairing takes place, right? So here the chromosomes, they pair with each other, and these homologous chromosomes, they are also known as paired chromosomes. And the process of pairing of these chromosomes with each other is known as synapsis. There is also presence of a complex which is known as synaptonemal complex. And this synaptonemal complex is what? The chromosome synapsis is accompanied by the formation of complex structure. So when the synapses, as the chromosomes are pairing with each other, a complex form is produced, a complex structure is produced, and this complex is known as synaptonemal complex. This complex is formed by pair of synapsed homologous chromosomes and is called bivalent or tetravalent. So as this complex is pairing with the homologous chromosomes which are occurring at the synapse, right? For this reason, they are said to be bivalent, and if the two pair of chromosomes are occurring, then it is said to be tetravalent. So if one pair, if it occurs in one pair, then it is known as bivalent, and if it occurs in two pair, then it is tetravalent. So this is about the second phase of the prophase 1. The third phase is pachytene, where 4 chromatids of bivalent chromosome appears at tetrads. So from these 4 chromatids, right, the 4 chromatids, they are bivalent chromosomes, right? So two chromosomes are present in order to form the 4 chromatids. So as 4 chromatids are formed, for this reason they appear as a tetrad. Tetrad word is used for when it is present 4. Next, the appearance of recombination nodules is observed. So in the pachytene phase, the recombination, that is recombination which is taking place of the chromosomes, is going to be seen in the pachytene phase. Recombination nodules are the site at which crossing over occurs. Now see here, recombination means something which is again combining, right? So already the pairing has taken place, but again the chromosomes are going to be crossed with each other. So if we have two colored chromosomes, a pink color and a blue color, right? Even though the pairing has taken place, that is one strand is of the pink color and second strand is of the blue color, right? So we have two different strands. But what happens when the recombination takes place? Some part of the pink colored chromosome goes into the blue color, and the some part of the blue colored chromosome goes into the pink. So now we have this process of passing the particular, or we can say the crossing over, which is taking place between the two chromosomes, that is known as recombination, right? So this recombination nodules, these are the sites where the crossing over takes place. And recombinase is the enzyme which carry out the crossing over, right? So if it is asked in MCQ that which enzyme is responsible for carrying out in the crossing over, then that is recombinase, right? Now, crossing over is the exchange of genetic material on two chromosomes. So, as I said, that it is going to pass the genetic material from one chromosome to the next chromosome. So we have two strands over here. So from one strand, half of the or few part of the genetic material is being exchanged with the next or the adjacent chromosome. Same way, from the next chromosome, some of the genetic material is being passed to the previous chromosome. So this way the crossing over takes place. So recombination of chromosomes is completed in this stage. So in this particular stage, that is packaging stage, the recombination of the chromosomes is completed. Next is the diplotin phase, and this diplotin phase, in here what happens, the synaptonymal complex, they separates in this phase. So here what happens, whatever complex which was very complex in nature, here this complex gets separate, right? And recombined chromosomes of the tetrad separate from each other except at the sites. So wherever the crossing over has taken place, except those places, other part of the chromosomes, they start for the recombination part. And this egg-shaped structure is known as chiasmata. So what is chiasmata? Where the crossing over, whenever the crossing over takes place, so it forms a egg-shaped structure, and that is known as chiasmata. The fifth phase of the prophase one is dikinesis, which is nothing but the terminalization of chiasmata. So nothing but the stopping of this egg-shaped structure, that is for dikinesis. So chromosomes are fully condensed. That means the DNA or the genetic material which is present, it is fully condensed. That is, it is compacting with each other to form the structures which are chromosomes. The meiotic spindle is assembled to prepare the homologous chromosomes for separation. So here what happens in the dikinesis is, when the chromosomes are fully condensed, the meiotic spindles are arranging themselves to prepare the homologous chromosomes for the separation. So by the end of the dikinesis, the nucleolus and the nuclear envelope disappear. So this is the phase of the prophase 1, where the nuclear envelope, that is the nuclear membrane, as well as the nucleolus, those two of them, they degenerate, right? Or we can say they disappear. And due to this, what happens, it starts to disappear. So when the metaphase is entered, right, at that time the nuclear membrane is degenerated, and for this reason every genetic material and all the things are dispersed inside the whole medium of cytoplasm. So this is about the prophase 1 of meiosis 1. Now we are going to see about the metaphase 1. So what happens in the metaphase 1? The bivalent chromosomes align on an equatorial plane. So just same as the mitosis, right? Here also the chromosomes, now they are going to assemble themselves on the equatorial plane. So they are going to be arranged in one single line. And the microtubules, they are from the opposite poles, they start forming the spindles, and they are at these spindles are attached to the homologous chromosomes. So this is the second phase of the meiosis 1, and that is metaphase 1. The third is the anaphase 1. In the anaphase 1, the homologous chromosome separates while sister chromatids remain associated at their centromeres. So here what happens now? The separation, just as the mitosis, right? Here also the chromosomes are now joined. So from centromere, the two sister chromatids, they separate from each other, and what happens? They start moving towards the poles. The fourth phase is the telophase 1, and here the nuclear membrane and the nucleolus reappear. So as you know that now the two cells are about to divide, for this reason the nuclear membrane and the nucleus starts to reappear, and this phase is followed by cytokinesis, and cytokinesis is nothing but the division of the two cells from one, right? And interkinesis is what, the stage between the two meiotic division, right? The cytokinesis is what, the cell division which is taking place, right? And the interkinesis is what, the two meiotic, the stage which is present between the two meiotic division is known as interkinesis. And this interkinesis is followed by the prophase 2. So here at the cytokinesis, so here upon carrying out the after the nuclear membrane and the nucleolus reappear, what happens? The cytokinesis is taking place. So what is cytokinesis? So it is dividing one cell into two daughter nuclei, right? So here the two daughter cells are produced. So the product of the first meiosis is two cells, right? The two sister chromatids. Now, after the interkinesis, the this is the figure you can see here. This is the prophase one, where the assembling of the chromosomes takes place. This is the metaphase, where it is uh, occurring, or it is they are present in on the equator. In the anaphase, they separate from the centromere and they start moving towards the poles. And in the telophase, finally the two cell divides, right? So now that was about the meiosis 1. Now we are going to see about the meiosis 2. So in the meiosis 2, it resembles a normal mitosis. So this meiosis 2 is just same as the exact mitosis. So the difference between the mitosis and meiosis is just about the meiosis 1. And that too, in the meiosis 1, the prophase 1 is quite complex in comparison with the mitosis prophase, right? So here what happens? This meiosis 2 is exactly what happens with the mitosis. So it's just same as mitosis. So in the prophase 2, it initiate immediately after cytokinesis. So just the two cells, when the two cells divide, suddenly the prophase, it enters into a phase which is known as prophase 2. So the here again the nuclear membrane starts to disappear and the chromosome again become compact. That means they condense and they form the structures which are known as chromosomes. Second is the metaphase 2. In this stage, the chromosomes align at the equator. So now what happens? When the nuclear membrane disappears, everything is dispersed inside the cytoplasm. And for this reason, the chromosomes, they set into a line and on the equator. So in the middle central line, the chromosomes get assembled, right? The microtubules, from the opposite poles of the spindle get attached to kinetochores. So the chromosomes, they are going to attach to the spindle fibers, and through the spindle fibers they are steady at the middle portion, or we can say the equator. Next, and the third most phase is anaphase 2, where the splitting of the centromere of each chromosome takes place, right? So the centromere of the chromosome splits, and for this reason, the two of the sister chromatids, they starts to move towards the two opposite poles. So now the splitting has taken place, so the chromosome starts moving towards the two opposite poles. And the last and the final phase of the meiosis is the telophase 2, where the chromosomes of two groups get enclosed by nuclear membrane. So when the nuclear membrane starts to reappear, the chromosomes are enclosed inside it. And upon carrying out the cytokinesis, so again the cell division takes place. So from two cells, another two cells are produced, and it results into four haploid daughter cells, right? So this is the figure where you can see here. This is the prophase 1, and it forms the two cells, and this is the this is the meiosis 1, right, where I'm showing the cursor, and it forms the two cells. Same way, this is the meiosis 2, where again it forms this two cells. So at last you have four cells, 1, 2, 3, and this is number 4. So at last or at the end of the meiosis 2, we do possess four haploid daughter cells. Now we are going to see the significance of meiosis, where the conservation of specific chromosome number of each species is achieved across generation. So now we can through meiosis we can understand that how the specific, what exactly the specific chromosome number is of a particular species, right? And how many, after how many generations, where if any mutation or if evolution has taken place, then what is the exact change which has taken place. So for the conservation of the those specific chromosome number is being carried out with the use of meiosis. Also, it increases the genetic variability, that is the evolution which is taking place, and that is also due to the meiosis. variations lead to evolution. So none another, if it increases the genetic variability, so variation takes place, and through the variations the evolution is occurring. So these are the 3 points of the significance of the meiosis. Here we complete this chapter. I am hope you are very clear with today's topic, and I hope you are you have already understood about the two types of main cell division, that is mitosis and meiosis. So I hope to see you again. Thank you.